fatty acid is a carboxylic acid with a long hydrocarbon chain. Fatty acids are important dietary sources of fuel for animals and are important structural components for cells. Triacylglycerol are the main constituents of the body fat in humans and other vertebrates. In this lecture, we will cover the following topics. We will begin with fatty acids. Fatty acids have three features. First is amphipathic. The carboxyl group is hydrophilic and hydrocarbon is hydrophobic. Second, most fatty acids in plasma appear as fatty acid esters contained in lipoproteins. Third, fatty acids may be saturated, no double bonds, or unsaturated with one or more double bonds. Double bonds usually 3 carbons apart. For example, these double bonds are 3 carbons apart. Following standard method, the carbon from the carboxyl group is designated as first carbon. The next carbon is second carbon, and third carbon, and so on. Following alternative method, the first carbon following the carboxyl group is alpha carbon, and the second carbon following the carboxyl group is beta, and gamma, and so on. The last carbon in the chain is called omega carbon, and therefore the third from the end is called omega minus 3 carbon. There are 3 methods to name a fatty acid. First is to use common name. For example, arachidonic acid, linoleic acid. Second is delta system. Following this system, first number describes the total number of carbon. Second number describes the total number of double bond, followed by a delta sign, and position numbers of double bonds. For example, here is linolenic acid. It has 18 carbons and 3 double bonds, and the 3 double bonds are found at position 9, 12, 15. So therefore, linolenic acid can be described as 18:3 delta 9, 12, 15. Third method is omega system. Following this system, the first number describes the total number of carbon, second number describes the total number of double bonds, followed by the position number of the first double bond from the omega end. For example, linolenic acid can be described as 18:3 omega 3 in parentheses. Most fatty acids in humans have 16, 18, or 20 carbons, but several fatty acids with longer chains occur principally in lipids of the nervous system. Fatty acids with fewer than 6 carbons are short-chain fatty acids. Those with 6 to 12 carbons are medium-chain fatty acids. Fatty acids with 13 to 21 carbons are long-chain fatty acids. Those with more than 22 carbons are very long-chain fatty acids. 2 essential fatty acids, linoleic acid and linolenic acid, have 18 carbons. Several special fatty acids are important intermediates of energy metabolism. For example, these 2 compounds contain alpha keto group, Hyaluronic acid is a degradation product of alanine, cysteine, and serine. Alpha-ketobutyric acid is a degradation product of methionine and threonine. These three compounds contain two carboxyl groups. They are called dicarboxylic acid. These first two are intermediates of TCA cycle, and this compound is a product of omega oxidation, which we will see later in this lecture. These two compounds contain two carboxyl groups plus one keto group. They are degradation products of amino acids and components of TCA cycle. Almost all fatty acids in humans are straight chain, but phytanic acid is an exception. Here is a phytanic acid. It contains methyl side chain. It has clinical correlation. Inability to degrade phytanic acid causes Refsum disease, which we will cover again later in this lecture. Now let's move to part 2. Dietary lipids can be roughly divided into 6 classes: fatty acids, triacylglycerol, phospholipids, vitamins, steroids, and glycolipids. Their structures are shown on the right. Hydrophobic portions of the molecules are highlighted in orange. Most lipid digestion takes place in the small intestine. Main enzymes are derived from pancreas. Emulsification in duodenum by bile salts and segmentation contractions in the intestine increase the surface area of lipid particles to facilitate digestion by pancreatic enzyme. Here is triacylglycerol. Two fatty acids are removed from position 1 and position 3 by pancreatic lipase. The result is 2-monoacylglycerol. Here is phospholipid. Two fatty acids are removed from position 1 and position 2 by 2 lipases. The result is glyceryl phosphorylcholine. Here is cholesterol ester. One fatty acid is removed from position 3 by cholesterol esterase. Cholesterol is released. The enzymes and their actions are summarized on this slide. Here, co-lipase help pancreatic lipase in digestion of TAGs. Phospholipase A2 and lysophospholipase are the two lipases that cleave fatty acids from phospholipid at C2 and C1 position, respectively. Short-chain and medium-chain fatty acids are absorbed directly into enterocytes. Long-chain fatty acids and bile salts form micelles. When micelles approach brush border of mucosal cells, long-chain fatty acids are absorbed. After long-chain fatty acids enter the cell, they are resynthesized into TAG, phospholipid, and cholesterol ester, and packed into chylomicrons. Bile salts are absorbed in the terminal ileum. Here is further description of resynthesis of triacylglycerol. Long-chain fatty acids are first activated by attaching them to coenzyme A. Fatty acids are then transferred first to monoacylglycerol, giving rise to diacylglycerol. The same enzyme transfers a second fatty acid to diacylglycerol to form triacylglycerol. TAGs are packed into chiral microns for delivery. So, what is chiral micron? Nascent chiral microns are large particles containing TAGs, cholesterol ester, and fat-soluble vitamins in the middle, and cholesterol, phospholipid, and apolipoproteins in the outer shell. Nascent chiral microns are created in the ER of enterocytes. Two proteins essential for the assembly of chiral microns are mycosomal triglyceride transfer protein, or MTP, and apolipoprotein B48. Here is clinical correlation. First, lipid malabsorption. It results in loss of lipids, fat-soluble vitamins, and essential fats, with steatorrhea. Second, anti-obesity drug orlistat inhibits gastric and pancreatic lipase, decreasing fat absorption. This is how this drug can help lose weight. Third, A-beta lipoproteinemia is caused by mutation in MTP. Mutation results in deficiency in the apolipoproteins B48 and B100. ApoB48 deficiency blocks absorption of fat and fat-soluble vitamins from food. Key findings include absence of chylomicron, VLDL, and LDL in the blood. Symptoms include failure to survive, diarrhea, and fatty, foul-smelling stools with acanthocytosis. Now let's move to part 3. Biosynthesis of fatty acids occurs mainly in the liver, lactating mammary gland, and to a lesser degree in adipose tissue and kidney. Inside the cell, fatty acid synthesis occurs in the cytoplasm. It uses Acyl-CoA in carbon dioxide as precursor and uses NADPH as a coenzyme. Fatty acid synthesis consumes ATP. Fatty acid synthesis can be further broken down into 3 separate pathways. First, synthesis of palmitate from Acyl-CoA. And second, palmitate can be elongated. And third, fatty acids can be desaturated to create double bonds. In starvation, fatty acids is transported into mitochondria, where it is oxidized to Acyl-CoA. A rising level of Acyl-CoA promotes gluconeogenesis. In the fat state, ATP is high in the mitochondria, and it inhibits isocitrate dehydrogenase. Acetyl-CoA condenses with OAA to form citrate. Citrate is transported to the cytoplasm, where citrate is converted back to OAA and acetyl-CoA. Acetyl-CoA is then used to synthesize fatty acids. Therefore, acetyl-CoA is a key intermediate between fat and carbohydrates metabolism. After acetyl-CoA is transported into cytoplasm, OAA needs to be recycled. To recycle OAA, we will need 2 additional enzymes. First one is cytosolic NAD+ dependent malate dehydrogenase. And the second enzyme is NADP plus dependent malic enzyme. First enzyme converts OAA to malate. Second enzyme converts malate to pyruvate. Pyruvate is then transported into mitochondria to be converted to OAA. Here, the reactions catalyzed by malic enzymes produce 1-NADPH. This reaction provides an alternative source of NADPH besides HMP shunt. The major enzyme that synthesizes palmitate from Acyl-CoA is fatty acid synthase. Fatty acid synthase catalyzes the 7 different reactions, whereby 2 carbon units originally from monomer CoA are linked together. All 7 steps occur in the cytoplasm. The enzyme complex can exist as both a monomer and dimer. The dimeric form is a fully functional form of the enzyme. Each step requires loading 1 monomer CoA onto the enzyme. Monomer CoA is synthesized from acetyl-CoA by acetyl-CoA carboxylase, or ACC. This step consumes 1 ATP. ACC is a rate-limiting enzyme in fatty acid synthesis. The overall synthesis of palmitate from acetyl-CoA requires 7 ATP and 14 NADPH. To initiate fatty acid synthesis, we will need to load 1 acetyl-CoA onto one arm and 1 monomer CoA onto another arm of the fatty acid synthase. This is accomplished in 3 reactions. First, acetyl-CoA carboxylase synthesizes 1 monomer CoA. Second, monomer CoA is loaded onto one arm of the enzyme. Third, acetyl-CoA is loaded onto another arm of the enzyme. Here, acetyl-CoA carboxylase, like pyruvate carboxylase, use biotin as coenzyme. This reaction consumes 1 ATP. This is fatty acid synthase. This is phosphopendiphene arm. This arm accepts every new monomer CoA. Black dots represent the second arm that holds the glowing fatty acid chain temporarily. It is on the pendiphene arm where condensation reaction occurs. This reaction adds 2 carbon units to the fatty acid chain. The pendiphene arm is very reminiscent of A sites in ribosome. The next 3 steps are to reduce the carbonyl group, following a classic mechanism in organic chemistry. First step is to reduce the carbonyl group to hydroxyl group. Second step is to eliminate a water molecule. Third step is to reduce the double bond to single bond. In step 1 and step 3, each consume 1 NADPH. Now, this fatty acid group is ready to condense with another monomer group. Fatty acids are often modified. Modification include chain elongation and desaturation. Elongation occurs primarily in membranes of ER and involves the addition of monomer CoA to a fatty acid. Dehydrogenation also occurs in the ER by fatty acyl-CoA desaturase. This enzyme introduces double bond at position 4, 5, 6, or 9. Because linoleic acid and linolenic acid have double bonds beyond position 9, they are essential fatty acids. The fatty acid desaturase system is an electron transport system in the ER that involves three proteins. One is fatty acyl-CoA desaturase. Second is cytochrome B5. And third, NADPH cytochrome B5 reductase. For example, the single bonds in this fatty acid is oxidized to double bond, while oxygen is reduced to water. This reaction requires oxidation of cytochrome B5. To recycle cytochrome B5, cytochrome B5 reductase is oxidized. To recycle cytochrome B5 reductase, NADH is oxidized. Acetyl-CoA carboxylase is subject to allostatic regulation. Citrate activates acetyl-CoA carboxylase. Long-chain fatty acyl-CoA such as palmitate-CoA inhibit this enzyme. Glucagon and epinephrine activate a G-protein coupled receptor pathway, which activates cyclic AMP-dependent protein kinase A. Protein kinase A phosphorylates and activates AMPK kinase. AMPK kinase then phosphorylates and activates AMPK. Okay, AMPK is shown here. AMPK then phosphorylates ACC and make it inactive. AMP and metformin both activate AMPK. Therefore, these two chemicals inhibit fatty acid synthesis. Opposite to glucagon, insulin promotes dephosphorylation and activation of ACC. Long-term regulation involves control of transcription of two genes, especially high carbohydrate, fat-free diet and insulin increase transcription of ACC and fatty acid synthase genes. Diet and insulin first increase transcription of SREBP1. It stands for steroid regulatory element binding protein 1, a member of transcription factor. SREBP1 increases transcription of ACC and fatty acid synthase gene. On the other hand, high-fat diet, fasting, and glucagon decrease transcription of ACC and fatty acid synthase genes. They do this by decreased transcription of SREBP1 gene. Synthesis of triacylglycerol begins with glycerol 3-phosphate. Acyl transferase transfer the first acyl group from acyl-CoA to the C1 position of glycerol. Another acyl transferase transfers the second acyl group to the C2 position. Phosphatase comes in and removes the phosphate group. Then the third acyl transferase transfers the third acyl group to the C3 position, giving rise to triacylglycerol. In both liver and adipose tissue, glycerol 3-phosphate is mainly derived from dihydroacetone phosphate from glycolysis. In the liver, glycerol 3-phosphate can also be made from glycerol by glycerol kinase. This reaction only occurs in the liver. Now let's move to part 4, Fatty acids in the form of TAG serve as the body's major fuel storage reserve. During starvation, hormone-sensitive lipase is the first enzyme to release fatty acids from the storage. Epinephrine activates hormone-sensitive lipase by phosphorylation. Hormone-sensitive lipase release fatty acids from TAG at position C1 and C3. Then monoacylglycerol lipase release the last fatty acids. Fatty acids are bound to albumin when circulating in the blood. The major pathway to break down fatty acids is a mitochondrial pathway called beta oxidation. This is beta oxidation. First task is to move fatty acids from the cytoplasm to the mitochondria. Short-chain and medium-chain fatty acids can cross the mitochondrial membranes easily. Big challenge we face is how long-chain fatty acids cross the inner mitochondrial membrane. Long-chain fatty acids are transported across the impermeable inner mitochondrial membrane via the carnitine shuttle. This is the outer membrane. This is the inner membrane. To cross the outer membrane, fatty acids need to be activated by attaching to coenzyme A. To cross the inner membrane, it requires carnitine shuttle. This involves three enzymes. First, fatty Acyl-CoA need to switch to carnitine shuttle. This is done by CPT1. CPT1 stands for carnitine palmitoyl transferase 1. The product, acylcarnitine, can now cross the inner membrane through translocase. Once inside the matrix, acylcarnitine is switched back to fatty Acyl-CoA. This is done by CPT2. Before crossing these two membranes, fatty acids need to be activated. Activation of fatty acids is catalyzed by Acyl-CoA synthases. This reaction occurs in outer mitochondrial membrane and ER membranes. It takes two steps. First, fatty acids is transferred to a nucleotide. Here is a nucleotide ATP. Fatty acids arrive here. Second, fatty acids is switched to bond with coenzyme A. Now fatty acids is activated. Activation of fatty acids consume 1 ATP. Regulation of carnitine shuttle occurs at CPT1. Specifically, malonyl-CoA inhibits CPT1. High level of malonyl-CoA signifies the states of fatty acid synthesis, thus preventing the entry of long-chain acyl groups into the mitochondrial matrix will ensure the newly made palmitate cannot be transferred into the mitochondria and degraded. Carnitine is primarily found in meat products. Carnitine can also be synthesized from lysine and methionine in the liver and kidney, but not in muscle. Therefore, muscle depends on carnitine provided by the liver and kidney or the diet. Here is clinical correlation. Carnitine deficiency. Carnitine deficiency results in decreased ability of tissues to use long-chain fatty acids as a fuel. Primary carnitine deficiency may include deficiency in carnitine shuttle, such as CPT1 deficiency and CPT2 deficiency. CPT1 deficiency affects the liver, where an inability to use long-chain fatty acids for fuel impair liver's ability to synthesize glucose during fasting. This can lead to hypoglycemia. CPT2 deficiency occurs primarily in cardiac and skeletal muscle. Symptoms range from cardiomyopathy to muscle weakness with myoglobinemia following prolonged exercise. Carnitine deficiency may also be caused by deficiency in renal tubular reabsorption of carnitine, mutation in carnitine transporter OCTN2, leading to decreased level of plasma carnitine level. This type of deficiency is called carnitine transporter deficiency. Third, deficiency in membrane carnitine transporter that is involved in carnitine uptake by cells. As I mentioned to you earlier, short-chain and medium-chain fatty acids can cross the inner mitochondrial membrane without carnitine shuttle. Once inside the matrix, fatty acids undergo beta oxidation. It consists of 4 reactions involving the beta carbon right here. Beta oxidation follows a classic mechanism in organic chemistry. First step is the oxidation step, in which single bond between beta and alpha carbon is converted to a double bond. Second step is hydration step, giving rise to a hydroxyl group at the beta carbon. Third step is another oxidation step in which hydroxyl group is converted to a keto group. The fourth step is to break right next to the beta carbon using coenzyme A. The result is Acyl-CoA and shortened fatty Acyl-CoA. Here we have 2 oxidation steps. First oxidation step is catalyzed by Acyl-CoA dehydrogenase. It generates 1-FADH2. Second oxidation step is catalyzed by 3-hydroxyacyl-CoA dehydrogenase. It generates 1-NADH. Since palmitate needs to go through beta oxidation 7 times before it's completely degraded, beta oxidation of palmitate yields 7-FADH2 and 7-NADH, which are equivalent to 35 ATP. Here is clinical correlation. Acyl-CoA dehydrogenases comprise a family of chain-length specific enzymes, including very long-chain, medium-chain, and short-chain Acyl-CoA dehydrogenases. Among them, medium-chain fatty Acyl-CoA dehydrogenase deficiency is most common. It causes a decrease in fatty acid oxidation, resulting in hypoketonemia and severe hypoglycemia. Medium-chain Acyl-CoA dehydrogenase has been implicated as a deficiency in up to 10% of infants who die from sudden infant death syndrome. Regulation of fatty acid degradation occurs at 2 levels. First, it occurs at lipolysis. During starvation, glucagon and epinephrine increase protein kinase A-mediated phosphorylation, activates hormone-sensitive lipase. During fat state, insulin promotes removal of phosphorylation from hormone-sensitive lipase, keeping it inactive. Second, it occurs at beta oxidation. Malonyl-CoA inhibits CPT1, and it prevents the entries of long-chain fatty acids into the mitochondrial matrix. Acetyl-CoA to CoA ratio also plays a role. As the ratio increases, the CoA requiring thiolase reaction decreases, and therefore beta oxidation decreases. So far, we have seen how palmitate is degraded through beta oxidation. palmitate has 16 carbons, and we only need to run beta oxidation 7 times to completely break down palmitate. What happens if we encounter an odd-chain fatty acids? Beta oxidation of odd-chain fatty acids will eventually lead to 3 carbons propionate-CoA. To break down propionate-CoA, we add 1 carbon to it to make it a 4-carbon fatty acid CoA. This reaction is catalyzed by propionate-CoA carboxylase. Very similar to acetyl-CoA carboxylase, it requires biotin as coenzyme. The resulting methylmalonyl-CoA is not straight. 2 steps of rearrangement leads to succinyl-CoA. Succinyl-CoA can be degraded through T-cell cycle. Here, one enzyme needs our attention. Methylmalonyl-CoA mutase require vitamin B12 as coenzyme. Beta oxidation also occurs in peroxisome. The primary function of peroxisomal beta oxidation is to shorten very long-chain fatty acids so they can be degraded in the mitochondria. The presence of methyl group at the beta carbon in phytanic acid prevents beta oxidation. To break down this phytanic acid, we have to use an alternative pathway. That's alpha oxidation. Alpha oxidation occurs in peroxisome. It requires oxidation at alpha carbon by phytanoyl-CoA alpha hydroxylase. This enzyme adds a hydroxyl group to alpha carbon. Then a lyase cleaves the terminal carboxyl group together with coenzyme A. The products can now be attached to CoA and go through beta oxidation. Refsum disease is most commonly caused by phytanoyl-CoA hydroxylase deficiency. In Refsum disease, buildup of phytanic acid primarily affects nervous system. Patients present with neurologic damage, cerebellar degeneration, and peripheral neuropathy. Omega oxidation occurs in the smooth ER of liver and kidney. Omega oxidation involves oxidation of medium-chain fatty acids from the omega end. The result is dicarboxylic acid. When beta oxidation is defective, a typical finding is buildup of dicarboxylic acid. Now let's move to part 5. Ketone bodies include acetoacetate, 3-hydroxybutyrate, and acetone. Synthesis of ketone bodies occurs in the mitochondria of liver cells. It involves 3 key enzymes. The first step, 2-acetyl-CoA combine to make acetyl-acetyl-CoA, 4 carbon. Second step, add 1 more acetyl-CoA. The result is HMG-CoA, 6 carbon. This step is catalyzed by HMG-CoA synthase. Third step, removes 2 carbons along with coenzyme A. The result is acetoacetate. This is 1 ketone body. This reaction is catalyzed by HMG-CoA lyase. Acetoacetate can be reduced to 3-hydroxybutyrate by using NADH as a reducing agent. This reaction is catalyzed by 3-hydroxybutyrate dehydrogenase. This reaction is controlled by NAD plus and NADH ratio. This is very similar to conversion between pyruvate and lactate. This slide highlights first two reactions in ketogenesis. This slide highlights the third reaction catalyzed by beta-hydroxybutyrate dehydrogenase. Again, this reaction is very reminiscent of the reaction by lactate dehydrogenase. Ketogenesis is regulated at three crucial steps. First, at lipolysis, by controlling hormone-sensitive lipase. Second, at entry to beta-oxidation, by controlling CPT1. Third, at the TCA cycle, specifically at isocitrate dehydrogenase. High NADH inhibits isocitrate dehydrogenase. It promotes ketogenesis. Low NADH promotes TCA cycle. Ketogenesis is reduced. Ketone bodies generated by the liver can be used as fuel by peripheral tissues, including the muscle and the brain. Use of ketone bodies requires transfer of coenzyme A from Acyl-CoA. This reaction is catalyzed by Acyl-CoA 3-oxaloacid-CoA transferase, or SCOT. Liver lacks this enzyme. As a result, liver synthesizes ketone bodies but cannot use them for fuel.